This video reviews the FDA's regulatory framework for botanical drugs, focusing on 4 approved case studies. We will explore the chemistry, manufacturing, and controls, or CMC, patterns that enabled their approval under the FDA's guidance for industry. The botanical pathway emerged through decades of drug discovery shifts. Key milestones include the first, guidance edition in 2004, and the second in 2016, which codified the totality of evidence framework. Approximately 150 botanical applications are currently active in the pipeline. We will examine 4 FDA-approved botanical drugs: Verigen, Mytesi, Nexobrid, and Filsuvez. Each case study reveals how the unique CMC challenges of complex botanical mixtures were solved using the totality of evidence framework. Verigen, approved in 2006, was the first botanical NDA. It is a green tea extract ointment for treating genital warts. Key advantages included a pre-existing safety profile from global green tea consumption and an unambiguous clinical endpoint of complete wart clearance. The core CMC challenge for Verigen was the irreducible compositional variability in catechins from the Camellia sinensis source. Additionally, no direct correlation existed between individual catechin levels and the clinical response, precluding traditional structure-activity relationship-based specifications. Verigen established the totality of evidence approach using 3 strategies: upstream control via cultivar restriction and GACP, specifications anchored to the composition of clinical trial batches, and dual dose testing demonstrating dose insensitivity, providing a robust clinical anchor. Mytesi, the first approved oral botanical drug in 2012, is a delayed-release tablet for HIV-related diarrhea. It leveraged centuries of traditional Croton Leclerc use for a safety foundation and addressed an urgent unmet medical need. A known mechanism of action enabled a key quality solution. Mytesi's complex oligomeric mixture had combinatorial diversity that exceeded the power of conventional analytics. This forced the FDA to formally escalate the evidentiary bar beyond what was required for Verigen's less complex composition. The Mytesi solution was the, quote, clinically relevant bioassay precedent. Quality assurance was grounded in a chloride channel bioassay, measuring therapeutic function rather than chemical composition. This was coupled with ecogeographic region restriction and clinical consistency across multiple doses and batches. Nexobrid, approved in 2022, is an enzymatic debridement concentrate derived from pineapple stems. It was the first botanical regulated as a biological product, filed under the BLA pathway, matching its complex enzyme mixture nature to the biologic framework's activity-based specification precedent. The challenge for Nexobrid was that what it does outruns what it contains. The complex mix of bromelain-related proteases meant individual contributions to selective eschar removal were undefined. Batch-to-batch consistency was also constrained by plant source variability. Nexobrid's quality was captured by function, not composition. Controlled processing established the, quote, process is the product paradigm. Proteolytic activity assays served as the primary functional quality specification, directly anchored to the quantitative eschar removal clinical endpoint. Filsuvez, approved in 2023, is a 10% oleogel of birch triterpenes for wound treatment in epidermolysis bullosa, an orphan rare disease. Advantages included the orphan drug framework, providing market exclusivity, and prior EMA approval leveraging the ES trial data. The CMC challenge for Filsuvez was multi-axis variability. Triterpene composition varies significantly with birch species, growing region, and harvest conditions. Furthermore, a single marker like betulin couldn't represent the full therapeutic profile. Filsuvez demonstrated the versatility of the totality of evidence framework across a new formulation modality. Its quality assurance involved quantitative triterpene profiling, controlled process parameters for the oleogel, and using European clinical data as a quality clinical anchor. This comparative synthesis shows the evolution of CMC strategies. We moved from Verigen's component-level quantitation to Mytesi's functional bioassay, Nexobrid's process is the product BLA, and Filsuvez's hybrid marker plus fingerprint approach. Each approval built upon the precedents of the last. 5 generalizable patterns define the governing architecture across all approvals. They start with upstream raw material control and include characterization calibrated to complexity, using bioassays as compensatory evidence, and clinical data as the ultimate quality anchor. The totality of evidence framework is the FDA's intellectual contribution, achieving the same patient protection standard as conventional drugs. Its 4 pillars are upstream raw material controls, partial chemical characterization, mechanistic bioassays, and multi-batch clinical data. Industry perception often misinterprets the few approvals as pathway difficulty. This is flawed. The true reasons are insufficient elapsed time, the financial profile of sponsors, and the mutual learning curve between industry and FDA reviewers. The totality of evidence acts as a de facto IP shield. Because quality rests on 7 interdependent elements, replicating the entire evidentiary system is difficult, potentially exceeding the barrier of a single composition of matter patent. Regulatory complexity stems from per-product review tailoring and cross-team review dynamics. The critical condition for success is whole team alignment on the totality of evidence principle to ensure predictability. The path to approval is navigable. Success depends on well-defined clinical endpoints, robust upstream controls, prior safety data, appropriate characterization, and strategic regulatory alignment. Consistent application of the totality of evidence principle is key.